Hi, my name is Antonia and I have prepared a presentation about metformin and glucose regulation or insulin kinetics. So, type 2 diabetes mellitus is characterized by insulin receptors to be less responsive to insulin circulating in blood. This is also called insulin resistance, and thus glucose uptake by cells is reduced while there is excess of insulin and glucose circulating in the blood. It is recommended for people suffering from type 2 diabetes to exercise, as during and after the exercise there is contraction-mediated glucose uptake into cells through blood-4. As well as during exercise, cells and mitochondria need more energy and are able to use glucose to be converted into ATP quickly. Moreover, with exercise, the number of mitochondria in the organism increase, which increase the level of glucose consumed, which overall help in reducing the glucose levels in the blood. Though there are medications developed such as metformin, which is known as the first-line pharmaceutical treatment for type 2 diabetes to help reduce the glucose levels if exercise is not enough. Metformin is being used to treat other diseases including cancer, obesity, liver diseases, cardiovascular diseases, and renal diseases due to its ability to exert different effects through different signaling pathways. Regarding type 2 diabetes mellitus and glucose levels influenced by metformin, metformin lowers plasma glucose by inhibiting gluconeogenesis and glycogenolysis, which decreases the glucose output by liver. Metformin suppresses liver glucose production through AMPK-dependent and independent pathways. Through AMPK-dependent pathway, metformin inhibits gluconeogenesis. This is done by two ways. The first is through AMPK-dependent activation of small heterodimer partner and inhibition of phosphorylation of CBP, which leads to suppressing the expression of gluconeogenic genes such as glucose 6-phosphatase or phosphoenolpyruvate carbokinase and pyruvate carboxylase. And the second way is the activation of AMPK, which leads to the inhibition of mammalian target or rapamycin complex 1, which also results in the suppression of gluconeogenesis. Also, metformin inhibits hepatic glucose production in AMPK-independent manner, where metformin weakens the ability of glucagon or inhibits mitochondrial glycerol 3-phosphate dehydrogenase, subsequently leading to an impairment of lactate utilization for gluconeogenesis. Recently, a study also demonstrated that metformin directly targets fructose 1,6 bisphosphatase 1, which is the rate-controlling enzyme in gluconeogenesis, and thus this inhibits the hepatic, which is liver glucose production as well. Also, metformin enhances GLUT1, mediated glucose transport into hepatocytes through activating insulin receptor substrate 2, which decrease the plasma glucose levels. And metformin decreases the liver glucose production as well as decrease glucose levels through increasing GLUT4, glucose transporter mediated glucose uptake in skeletal muscles, and absorption of glucose into the intestines. Metformin also stimulates glucagon-like peptide 1 release, which enhance the insulin secretion and lowering plasma glucose levels. Moreover, there have been some recent studies done on gut microbiota, which may be a target site of metformin, as shown by this dysbiosis of the gut microbiota in type 2 diabetes patients. The bacteroides fragilis in the gut decrease, which results in an increase in glycosadeoxycholic acid. The elevation of these acid levels suppresses intestinal FXR, which improves glucose tolerance. These are my sources, and thank you for watching. As we learned today, metformin is a very useful tool for people who struggle with high blood levels in glucose, but it's always still recommended for those people to exercise a lot to reduce those glucose levels, and then using supplements as metformin will be helpful, but they don't have to use this supplement or other supplements in that big amounts.